Welcome to the Science for the Public lecture series. Science for the Public is an organization committed to bringing science information and issues to the general public. Visit our website for our program listings and blog. Good evening, I'm Yvonne Stapp, and I welcome you to Contemporary Science Issues and Innovations. Tonight, we learn about the importance of microbes and why scientists know so little about them, despite the fact they are literally everywhere. Our guest is Dr. Slava Epstein, College of Science Distinguished Professor at Northeastern University. Dr. Epstein received his Ph.D. in microbial ecology from the Russian National Academy of Sciences in 1986. In 1989, when the Soviet Union collapsed, he gathered his family and emigrated to the U.S., arriving in Cambridge with no English, no job, no prospects at the time, and eventually he became famous anyway. This is an inspiring immigrant tale, much appreciated in a nation of immigrants, and he'll tell us more about it later this evening. But first, Dr. Epstein describes the universe of microbes and why about 99% of them remain unknown by what's called microbial dark matter. He explains why relatively few microbes can be grown in the lab, a serious concern at this time because of the urgent need for new bacterial candidates for the development of new antibiotics. Dr. Epstein and his colleague at Northeastern, Dr. Kim Lewis, are famous for an innovation called the iChip, which solved the problem of obtaining new microbe candidates for antibiotics and for the development of a breakthrough antibiotic they called Teixobactin. For this work, Drs. Epstein and Lewis received numerous honors. They were listed in the top 10 discoveries in 2015 by the United Press International, the top 5 most important medical developments of 2015 by the BBC, the top 100 discoveries of 2015 by Discover magazine. Dr. Epstein and Dr. Lewis were also in the top 100 global thinkers, Foreign Policy magazine. It is a very great honor to introduce Dr. Slava Epstein. Welcome, sir.
Thank you very much, Yvonne, for this introduction. It's a great pleasure to be here and a true honor.
And if we can start out with just getting a little background about the microbial world, I think it's not well understood. They may be all around us, but we don't hear a lot about it through the media and so on. So why aren't they well known? What's the big mystery?
Well, let me start from a simple analogy. Suppose you are a student of microbiology and you're taking a course and you have an exam to take tomorrow. You have a textbook, of course. You open it up and you see that 99% of the words are crossed out and all you have on a page is a few randomly distributed random words. What are the chances that you will learn enough of microbiology to pass the exam? Well, obviously the chances are nil. Yet that is exactly the situation in microbiology. Mm-hmm. What is relatively well known is about 1% of microorganisms, those at least from soils, even less so from aquatic environment. It's that tiny percent that is relatively well known because they have been cultivated in the lab.
Like E. coli.
Like E. coli. I wish I could tell you why this is so. I'm just pointing to the phenomenon referred to as microbial uncultivability often, which states that overwhelming majority of microbes simply refuse to grow in the lab. And in 150 years of microbiology, no one has been able to find a way to cultivate the rest.
So you have just this chosen few that will grow in the lab. So we all think that E. coli are the only microbes on Earth. So why don't they?
Well, Yvonne, if I knew the answer, we would have cultivated them. By the virtue of the fact that we are still not able to cultivate them, you can conclude that we don't know why. It's a pretty big mystery. It's a very intellectually teasing one.
Yeah.
It's one of the riddles that occupy my mind and minds of many people all the time. I confess I don't know the exact answer.
Okay, well, I can't resist asking, because you have pointed this out. Well, I guess it's known in microbiology. These are dense communities. They don't just stick with their own kind. One variety doesn't just stick with its own kind, but you're talking about like an ecological system in these microbial communities. Does that have anything to do with it? Do you see what I mean?
It's possible.
Okay.
The dependency of microorganisms on each other is very substantial. They tend to function in a way that is somewhat different, if not dramatically different, from communities of macroorganisms. Macroorganisms, say animals, they basically either compete with each other or eat each other, and that's about it. The diversity of the interactions is limited to a few interactions that are negative, really. It's dramatically different in microbial world. The byproducts of metabolism, call it waste, of one microorganism is a very valuable product to another. So nothing is wasted. It's used by a neighbor, and the waste products of the neighbor feeds another microorganism, and yet another microorganism coming back to the first, making a loop in which everything is cycled and recycled, and all those interactions are positive. Collectively, they can do the job that no one single organism can. It's not just a community. It's many people prefer to compare it with a superorganism, if you will.
And it acts like that, doesn't it? That it's all so completely interdependent, just like the organs in our bodies, and so if they don't operate on their own. So that seems like a possibility, and some can be isolated more readily than most.
It is a possibility. However, it has to be qualified a little bit. It is possible that one microorganism prefers to live in the presence of another one because they cooperate. Having said that, it's not exactly clear why we cannot create in the lab conditions that mimic the presence of the other, of one partner. That would make it possible to cultivate the first organism in pure culture in isolation if you can mimic the action of the neighbor. So why can't we do that?
Well, that's the other unresolved question. Take it. You have a lot of work to do, I guess, in this field. It's very, very rich. Now, why are microbes important then? I mean, so we can't get them, we can't identify them. You say this is dark matter. You don't even know what to call them if you can find them. Why are microbes so important?
It's hard to decide where to start. Perhaps I could start from a simple fact that composition of our atmosphere, the structure of soil, to a significant degree composition of the ocean is all the result of microbial activities. Not your activities, not my activities, but their activities. And they lived very happily together before a single multicellular organism. They learn how to cycle and recycle the material. All the elements, starting from carbon to nitrogen to phosphorus. What is waste for us, and then we look for ways to do proper waste management. For them is not a problem.
Isn't that interesting?
What is a problem for us, for them is an opportunity. Say oxygen in the atmosphere, it's entirely the product of microbial metabolism. Of course we can say it's plants today. Well, perhaps plants today contribute significantly, but plants have not invented that. They simply borrowed the abilities evolved in microorganisms and are using them now by, it is microbial in origin, the ability of plants to photosynthesize. It's microbial. Without microbes, there wouldn't be plants. Without plants, there would be still plenty of microbes. Everything from a chemical composition of our biosphere to cycling of the elements is, by and large, the work of microbes.
I understand geology also. Is that true?
That's
the rocks,
true.
everything on the planet needs it, so that in a sense the planet is alive, whether we are or not. It is a live thing, and it wouldn't function without the microbes.
I think it's a very fair statement.
Yeah. That's amazing, isn't it? And then we
is.
don't know anything about them. I mean, is it changing today, or are people in this field still kind of stuck with dealing repeatedly with just a few that they can identify in the lab?
I would say so.
Still has.
One lab or two labs or 22 labs cannot change the world. They can initiate the process, but they cannot change dramatically the landscape.
Okay. All right. So that we still have, in a way, a lot of problems. You have contributed a large solution in this, I believe. That one of the things, I guess, that has been driven the search for microbes has been antibiotics. And we have a crisis. Could you give us a little background on that? I'd like to talk about the iChip and what that does, because that captures these things well.
Yes, of course, the value of what you refer to as microbial dark matter is enormous for academia, but also very significant to the industry. And the need to get access to novel biological entities, to novel species, is great, because we sorely need sources of novel chemistry. This is especially evident in case of antibiotics and in view of the rise in antibiotic resistance. The discovery of novel antibiotics essentially stopped several decades ago. And this is very much connected to the fact that we can only cultivate a small portion of microorganisms. So the industry kept cultivating them and eventually started overmining them. So the discoveries became more and more rare, and eventually the pipeline dried to zero. That didn't stop microbes from fighting the existing antibiotics, developing defense against them. It has always been a race between the speed at which microbes can fight off our weapons and our developing new weapons. The moment we stop discovering antibiotics, the microbes got the upper hand. The antibiotic resistance is going up. The pipeline of novel antibiotics is basically zero, and it is a problem that results today in over 20,000 deaths in this country alone, probably around 50,000 between North America and Europe a year, and countless more, unnamed more, worldwide, and it's going through the roof.
Yes, there's a lot of concern because things like TB, they're so contagious and so on, are coming back ferociously. Let me ask about antibiotics, that you have all these types of microbes, but is it relatively few, the number that can be cultivated for antibiotics?
It is relatively few that produce antibiotics or antimicrobial entities that we can use. There are many organisms that produce substances that can kill or suppress growth of other microorganisms, but they tend to be so toxic to us as to become useless. It's not that they're absent as weapons in nature, they're present there, but they're useless for us. So you have to sieve through a number of, call them candidates, antibiotics, before you come up with something useful.
What would you look for? What are the features that would qualify it, like in Bactin, excuse me. What are the features of the microbe that would say, that would be good? You don't know.
I don't know. We don't know.
So they really don't know a lot about antibiotics either.
Correct.
Right. That's amazing because they've been at it for a very long time, and I guess it came up sort of by accident and still is a problem.
So in a way, it is basically a brute force approach. You cultivate, because antibiotics historically came principally from microbial sources. You cultivate microorganisms, you cultivate as many as you can, looking for those strains and those species that exhibit in the lab antimicrobial properties.
Right.
These become your hits for further investigation. But the start of the pipeline that begins with microbial diversity you cultivate in the lab and ends with a drug at CVS. It starts from a large effort that looks for organisms that may show useful properties, and this search is basically random. As I said, brute force approach to cultivate as much as possible.
Do you see any change coming down the line for that? Is there any possibility that you will, with some of the techniques you yourself have been developing, will be able to sort this out a little faster or easier?
Of course. For two reasons. One reason, I'm an optimist by nature, and the second, I hope to say I have a reason to be optimistic. Going to the second point, the reason, it appeared to us that in order to get access to microbial dark matter, this 99% or even more percent of organisms that we have never explored, we, meaning academia, industry, no one, in hope that they contain novel chemistry. It appeared to us at first that in order to get access to this microbial dark matter, you have to know the reasons why they don't grow in the lab, something we just discussed, but we don't know those reasons. It would appear it's necessary to know what limits microbial growth in the lab before you remove that restriction, allowing microorganism to grow. But this is catch-22. You cannot study microorganism until you cultivate it, and you cannot cultivate it until you know the properties. After a little bit of thinking, we realized that there may actually be alternative approach, that it may be possible to cultivate that, the properties of which are unknown, irrespective of your ignorance on the properties of this organism. I would like to briefly mention one thought experiment.
Yes.
It's very simple. If I take a cell from the environment, from the soil sample, a microbial cell, and put it into a petri dish, and if it grows, if it happens to be one of those that can utilize the resources of the petri dish, the nutrient medium, will start dividing, will form a colony. Of course, we call it cultivation.
Yes.
That's part one of the thought experiment. Part two, I take the very same cell and I move it to the lab, but halfway through that, I change my mind and I return the cell back to the environment without damaging it. So it goes back to the very same place where I initially sampled it from. What will it do? If it comes from a population successful at the time, the one that is active and growing, what does that cell do? Well, it will start growing just like its skin cells are growing, and it will form a colony, just like it did in part one of the experiment in petri dish. In part one of the experiment, we called the result cultivation. In part two of the experiment, shall we call that cultivation too? Well, it is cultivation. You sampled a cell, and then you found a place for it where it could form a colony. It is cultivation, isn't it? It is a useless cultivation for our practical purposes, because the result of that cultivation is mixed with growth of all the other organisms in nature. So what's the value of this thought experiment? Oh, there is a value. The value is in that it suggests very strongly that we had been trying to solve a non-existent problem. The problem is not cultivation. We just achieved the cultivation in nature. We know where to grow. We don't know why they're growing in nature. What this is so magic about the chemical soup in soil or in a pond or in the ocean that is uniquely capable to support microbial growth. That magic that is absent from petri dish. We don't know this, but we know where to find it. And that's in the original environment of the cell. So cultivation doesn't seem to be something in need to be developed.
I see.
The problem is different. The problem is how to separate that growth from other growths of other microorganisms.
Right.
But look, this is...
Can you do it?
But this is no longer biology. This is an engineering task. And once you reformulate the problem of how to get access to dark matter in this way, the solution becomes very easy. Going back to the thought experiment, what if, after I sampled the cell and took it halfway to the lab, before I return it to the environment, what if I put it into a small, call it a bag, with tiny, tiny, tiny pores in it, so small that this bacterium will not be able to leave that bag.
It is isolated.
So
okay,
it is
but
now,
it's in its community,
it is
but it's in a room by
isolated
itself.
from the environment.
Okay.
In a sense, it cannot escape this contraction.
Okay. All right.
But it can still communicate with the nature through those pores.
I see.
Now I return my bacterium to its natural environment,
Right.
not by itself, but inside that small bag.
Okay.
What will it do? Well, everything in nature, magic or not, will diffuse in. If the cell grows, the waste products will diffuse out. How will it ever know we had ever manipulated it? The idea is to trick the bacterium into thinking it is in its natural environment.
I see.
And it's not particularly difficult, I would think, because actually it is in its natural environment. And if it's capable of growth, in principle, it will grow inside that bag. And if it grows inside that bag, it will form a colony. And so after a period of incubation, a week or two, I will go back to this habitat and I will retrieve my bag, open it up, and I have a colony that I grew not knowing why. Well,
the thing is, you got the colony.
It is that simple, really.
Is that the principle behind the eye chip?
It is the principle behind the eye chip, which is nothing more than an array of those tiny diffusion chambers, each of which contains, on average, one cell. And this array goes into the natural environment of the cells, allowing them to grow. And we simply open it up, and all we need to do is to count colonies.
It's amazing that it seems so simple on the face of it, and then it's absolutely perfect. But I want to make sure the eye chip, it's full of holes for cells, but let me try to get this straight, is each one contains a cell, 400 holes or whatever it is, and they can communicate with their brethren, so to speak. Okay, so they're not isolated. They are in a sense, but they are not. Exactly, they're in two worlds simultaneously there. And this is how you came up with Teixobactin?
Yes,
sort of?
Precisely.
And
a project conducted by scientists at Biotech Pharmaceuticals, a small startup company that is our neighbor. It's actually 10-minute driving time from here. They conducted a research project supported by National Institute of Health to explore a possibility of eye chip producing novel microbial species, and those novel microbial species producing novel antibiotics. And they grew successfully 10,000 novel strains and species of microorganisms in a couple of years using this technology, eye chip. And some of those bacteria turn out to be fairly good producers of antibiotics, one of which happens to be teixebuctin, a novel antibiotic with fairly interesting properties.
Evidently, that it is, because I think one of the problems with antibiotics is that the bacteria develop resistance fairly quickly, but there was something special about this one that prevented that. Is that a rare quality among antibiotics? Is it something new that you discovered recently, or?
It is exceptionally rare property, and that explains in part relatively high interest to the molecule.
Yes, right.
It kills microorganisms in two ways, in two different ways, independently of each other. So imagine an antibiotic that has one target in the cell. When I say target, I mean a molecule or a structure. That molecule or that structure is affected by an antibiotic. The function of this structure is corrupted. The cell dies. Eventually, through mutational process, the cell, the progeny of the cell, will find a way to counteract the activity of antibiotic. The target will no longer be there, and that situation is what we call antibiotic resistance. That antibiotic resistance arises with a certain probability. It's small, but not zero. So sooner or later, it develops. Imagine now that your antibiotic kills the cell in two different ways. Then the cell has to modify both targets, both functions simultaneously. The probability of that happening in a single cell is one low probability multiplied by another low probability. It becomes infinitesimally small number. It's still not zero, but it's orders of magnitude lower than in case of antibiotics, and that's the majority of antibiotics, antibiotics that act upon only one target. That's the property of teixebuctin that makes it particularly interesting. The resistance simply does not arise at the frequency we can detect.
That is amazing. But it's also amazing that it is apparently still so rare to find one. I mean, you didn't just go out in the backyard and find that. I understand. I would like to turn also, you've done this work, and you know, at the moment there's a lot of concern about developing effective antibiotics, especially since we have overused so much the ones that we have that they're no longer effective. So good luck on that. But another area that you were interested in with this dark matter, I suppose, is that you could identify microbes gone or present on like in Mars, and I wonder if you'd tell us something. There's great interest every week. There's a report. Oh, we found something, but they really don't know. But you came up with a way to say we can identify them. Please take us there.
I'll be most happy to. So imagine my bag with tiny pores that is now empty. I do not put anything inside other than some gelling agent to support microbial growth when it happens. Otherwise, it's sterile, it's empty. So the pores are very small, microorganisms cannot get in. Only nutrients and other growth factors can diffuse in. Imagine now that I make a single opening, a single tunnel, a single pore that is larger than the other ones. The other ones, we can call them feeding pores because that's how food comes in. And the single one I'm making now, we can call it an entry pore because that's where microbes will get in. So if I have that bag and if I drop it into the environment of your choice, it can be ocean, it can be deep sea, under ice in Antarctica, or it can be Mars, for that matter. So what happens after that? If there are microbes, and on this planet microbes are everywhere, sooner or later a cell will find that entry pore. It will have the space to colonize. It will grow in because there are nutrients, the nutrients provided by diffusion, and will form a colony. Now, please tell me, where is microbiologist to know that? You see, with an eye chip or a petri dish, you need a microbiologist to sample cells, manipulate them, put into iChip, seal it, put into nature. Microbiologist has to be present. You can't use iChip on Mars because there is no microbiologist there.
You can't park one on Curiosity.
But that bag requires no microbiologist. You prepare it in advance and you throw it into a habitat you want to explore. And that habitat can be 10 kilometers deep underground or millions of light years away from us.
Right.
If there are microbes there, they will grow in.
Yeah.
How will we know that? Well, that's the second stage of development of the device.
Yeah.
We can build inside nanosensors that will register microbial activities, such as if the cell is respiring with oxygen, the concentration of oxygen will go down, and the sensor will make a record of it. If it, like us, respire CO2, there will be a sensor registering that. We will know that here is an organism that takes oxygen like us and respires CO2 like us.
Okay.
There may be any number of nanosensors which is an existing technology that will measure various aspects of metabolism of the cell inside.
Now, if you did this on Mars, if you put this, attached it to Curiosity and let Curiosity drop it off here and there, something or other on Mars, you would say you would not have to have a microbiologist right there. You could transmit this data back. So you need to deposit the instrument, the iChip-like thing, I guess, but you can catch that material back, and they don't have this with what they're looking for now as they investigate Mars right now.
This is correct. Most of the technologies used today to detect life extraterrestrially are based on chemical analysis of the samples, and the analysis are tailored to molecules that we believe are only produced by life as we know it. It's a very indirect way. It's the best way we have, but it's a rather indirect way of detecting life. And it's based on a significant number of assumptions.
Yeah.
The method we're discussing is in principle different. There we will use the most basic property of life, and that is the ability to propagate. And we will register such propagation. We can visualize that, and we can measure parameters of that growth. So it's very compatible with other ways to detect life and complements them, being in principle different.
I hope you'll be sending one off to Mars very soon.
Me too.
That's great. In our remaining time, since you have one of the most interesting, wonderful immigrant stories, I wonder if you'll tell us about it. I think a lot of people in this country will be greatly inspired, and I hope people coming into this country in the future will be, by this very moving story. So you were originally in the Soviet Union.
And let me go back to where we started. You described the arrival of my family and me as people who, well, didn't speak English.
Right.
Which is true. Didn't have any means, which is true, and no prospects, which is not true.
Well, I mean, prospects like somebody's going to offer you a job next week.
I would like to point out the very simple fact that the prospects were here. All that remained was to make sure you make a good use of those.
Right.
English can be learned, money can be earned, and our life for 30 years, and we arrived about 30 years ago, was under one umbrella. And this umbrella is that saying that where there is will, there is a way. We simply had to work hard, that's all.
But take us back to the Soviet Union time. We just need to remind people about the collapse. What happened leading up to that? I think that that was an issue by itself to motivate you to come to this country.
What happened in 1988, the year we left Russia, this was a combination due to combination of forces and factors. Russia, or USSR at the time, allowed people to immigrate and Jews to immigrate,
right.
And a good number of people, ourselves included, just simply use that opportunity to live for good, because the prospects were not there. The prospects were here.
Well, and on that, just if you would back up a little bit, I think you started out in physics and you were told, forget it, because?
This is true. While I did like biology since the time I remember myself, my heart for a period of time, and that was in elementary school, middle school, really belonged to astronomy. I was really, really, really passionate about it, and in many ways I still am. The most prestigious place to enter for further studies would be Moscow State University. I was told by a number of people, some of them were friends, others were relatives. As it turned out, one happened to be a professor at Moscow State University. I was told to not even think about it.
Because?
Because of the quota on Jews.
Yes, okay, so it's important that people understand this. There was a severe quota for Jews.
It was a severe quota on Jews.
Right, in case they are not aware of the hostility at that time, okay.
I have to say that at the time we were taking it for granted.
Of course,
because we knew no different.
Yeah.
The advice, and I eventually, in high school, I started paying attention to this advice. The advice was to switch from physics or / math, where the quota was the most severe, or was very well enforced. To something where there was the situation was a little bit more relaxed in that regard. Say biology. Biology was not seen at the time as something that had anything to do with defense or arms. It was more or less ideology free. It was easy to get into intellectual work through biology. I listened to the advice, never regretted it. But yes, the choice of biology was dictated essentially by political situation in Russia at the time. We're talking about 76 when I entered Moscow State University.
Okay, so then you could make a career, but I believe, were you a dissident at the same time there? Not that bad. You weren't a bad type or anything, no big threats or anything, but just...
No, I was not.
Okay.
It's a very honorable title, dissident. I don't think I have it.
It wasn't on the resume or anything like that, okay. All right, but in any case, it was uncomfortable, I take it, but I remember that time vividly when a lot of Jews from Russia were able to come here, and this is very different from World War II, after World War II, and so it has some resonance even today that, you know, this, that change, could other things change as well? In any case, you came over here, and since you had no language skills or you couldn't do what you were trained to do, what did you do? And this is a heroic thing.
Well, I did what I had to do and could do. And it turned out that for the first year or more likely two, what I could do was to paint fences and houses and repair roofs. And there was something I particularly liked, paving driveways with bricks, because you can make your own design. It was wonderful. It was tough but fair, and gave me ample time to study English, because you can just keep your earphones on you and
please tell us where you got the source of your English.
Well, from NPR and BBC.
Okay.
BBC was much harder.
Yes, because of the accent. Right. Yes.
and then miracles just happen. What at first appears to be a flow of noise, really. You cannot, or I couldn't even tell the difference between different words. It was one flow. Once exposed to it for long enough, plus the desire to understand, makes study of language relatively easy. I think it took a few months, maybe half a year, before realization came that what I was listening to was no longer noise, that I was actually listening to the news. Imperfect as my understanding was at the time, it was already there with no particular efforts. Really, just like a child learns.
Now, you got some language after a while, and this is just a huge thing for adults particularly, coming into especially professional people where you need a high level of language. And so you came, you did overcome things, you got some opportunities. As you say, the prospects were there. But so eventually you went to Northeastern, yes? And
yes, in parallel with this brickwork, I was also doing some volunteering in a microbiology lab at the University of Massachusetts in Boston, where I met wonderful people who were willing to help teach me how to write papers. I brought a lot of data, raw data from Russia to be published here, and they helped me big time to analyze the data, format it properly, write it down properly. It was without that work alone wouldn't do much good.
Right. I understand. It's not something.
And eventually, I
you do
became a postdoc at University of Massachusetts in
Right.
Boston, and in 1992, I believe, got my affiliation with Northeastern University, and has never left.
Right, and then the rest was history. You're all famous and everything now. And thank you for all the contributions in your new country that you have made. I hope that you will be contributing to the Mars discoveries here soon, and I hope that your work will continue. I guess it is in force now.
Once you taste the kind of research, such as antibiotic discovery, that brings or may bring immediate benefit to people, there is no way back. It is an enormously powerful driving force that just keeps you focused on that. Not the least because of the immediacy of the result. That the result will not be in 100 years or 1,000 years, but it may be actually tomorrow or in 5 years. But having said that, I cannot forget more academic questions and designing new ways to access dark matter, whether for the purposes of academia or the purpose of industry.
Just to understand that microbial world.
Tease apart the interactions, or maybe create something that may be useful to detect life elsewhere. This is also occupies part of it.
Dr. Epstein, thank you so much for talking with us and enlightening us and inspiring us.
Thank you very much. Thank you for having me.
Sure.
It was